Thank you. Probably most of you, or at least some of you, that have heard the term bipolarity or bipolar in the last years, it's definitely something different to what I'm gonna talk about. So apologies if you were expecting me talking about bipolar disorders in brainless animals. That's not gonna be the case. But this is a very interesting topic as well. So thanks for not going to the parallel session. Um, I'm John Soto. I work with polar jellyfish. I'm very interested in the ones that are bipolar. And what is bipolarity? Bipolarity is basically the presence of the same species or related species in both polar regions. Um, this phenomenon, this biogeographic phenomenon, is widely spread in the tree of life. It happens from virus to whales. There we have animals like the fulmars, they are bipolar. There's one species in the northern hemisphere, another species in the southern hemisphere. The right whales, the Cleone, the theropods are bipolar as well, and we have examples of jellyfish. For example, Periphylla is an example of a bipolar jellyfish. Um, if we track back the history of bipolarity, we have to go as early as nearly 200 years ago, where it already interested a lot of the scientific community. And James Clark Ross, he was by no means a biologist, but he was very interested in trying to figure out the connections between species that were very similar in both polar and subpolar regions. So he did many, many observations during his trips. Then Darwin, on the origin of the species, he already noted that some plants that inhabit in temperate areas, they can be also found in tropical areas, but at way higher altitude, so up in the mountains. And related with this, but instead of altitude going to deep waters, Berg realized that some temperate species can also be found in tropical waters, but only very, very, very deep. So we have these species that are found primarily in shallow or relatively deep waters in both hemispheres, but can also be found in very deep waters. And this is another type of bipolarity. Crane, he's a biogeographer and a paleontologist at British Antarctic Survey. He has published a lot on mollusks, especially based on the fossil record, and he has been able to define divergence time based on the fossil record for many mollusk lineages. So he was actually able to track the origin of this split of this diversification using the fossil record. But still, for many groups, there is a lot of gaps in the knowledge because there is not a lot of molecular data supporting this evidence. So we have a snapshot. We know that bipolar distributions occur. We know that there is one species in the north, another in the south, but we don't know how and when these distributions appear. Which is the origin of this peculiar distribution. So Alcon and Griffiths basically summarize all the literature on the topic, and they basically provide two hypotheses. Either it's through vicariance, in which we are in a glacial period, we have a cosmopolitan, widely distributed species, and then the warmer it gets, this species basically splits and goes northern and southern, and then we have a barrier that the species is not able to keep gene flow, and then it speciates, and we have basically two species, one in the northern hemisphere and another one in the southern. The other hypothesis is one species that it's distributed only in one of the hemispheres. We are in an interglacial period, so we are in a warm environment as we are now, and then the globe gets colder and colder and colder, and this northern or southern species is able to jump the equator, and then through founder effect, it's able to establish itself in the other hemisphere, and then we have the same, right? So we have two potential origins. There are more hypotheses, but broadly we have these two potential explanations, but again, it's a matter of how and when. So we don't know, and for this, we need to include either fossil record, molecules, or preferably both. We are lucky enough to have had Sofia Stepan-Jans within the Cnidaria Medusozoa Hydrozoa community. She has worked a lot on bipolar. She was super interested in bipolarity in Medusozoa and Hydrozoa particularly. And she identified 32 genera and 23 species that sensu lato could be considered bipolar. There's more genera than species because some genus can be considered bipolar, even though not all the species within the genus are bipolar. So she found several species, and she also suggested that there are some genera that could also be relict of ancient times, that they have be kept in polar regions, but they were a wider group in previous geological times. Recently, we reviewed the existing information about it, and we narrow it down to only the species that are strictly bipolar, because they are only in the poles, and most of them are absent anywhere else in the world. And but we are still lacking molecular data for most of them. So the question, at least for Hydrozoa, is still how and when these distributions arose. And here's when our project Pole to Pole came into the game. It's a project funded by European Commission under the program Marie Curie within the Horizon 2020. And we want to use Hydrozoa and their wider range of life cycle strategies to answer the question whether life cycle strategies have something to do with the time of diversification. Our hypothesis is that for those species that have highly dispersal capabilities, they should have kept somehow gene flow, and they should have an earlier split. So they should have kept gene flow not so long ago. And then for those species with very limited dispersal capabilities, such as benthic hydroids without a medusa stage, this split should have happened earlier in the time history. So we are going to the polar regions, collecting many samples. We are revisiting the species delimitation. We are working with both Sanger sequencing and next generation sequencing to see how many lineages we have. It is a matter of how many species do we have in there. Is there cryptic speciation in there? Are those species the same? And then the final goal is to be able to determine how and when this split occurred using calibrated trees. These are the models we are working with. We have selected them because they represent very well the different life cycle strategies. So on one hand, we have Buyonia. It's a benthic polyp. It doesn't have medusa stage. And it has, it's very peculiar because it self-recruits. The polyps are shedding the larvae into the water, and basically they settle a few meters away from the mother polyp. This is known for the species. So in principle, it's an example of very restricted dispersal capabilities. Then we have a benthic hydromedusa, a Trachymedusa Tychogastria polaris, which has been described from both poles. Then we have a group of species that have both polyp and medusa stage, the group Tychochina stegopoma, I will explain later. And then we have a representative of an holoplanktonic life cycle, Dimophias arctica, that, as the name says, it was reported originally from the Arctic, but we found it in the Antarctic as well. So I will briefly tell you a bit more about its case, and whenever we have results, I will show them to you. There's not much we can say about Buyonia. So far, we have some sequences, and it seems that it has a very peculiar position within the phylogenetic group it belongs. It's a Tubularidae, and we don't have much data about it, but it really seems it's like the sister group of all the other Tubularidae. If this is the case, it could confirm the hypothesis given by Stepan Jans that it could actually be a relict group from polar regions. It is a species that you find very often on moors and moorings in very deep waters. So if you ever see it, just give me a call because it's a very interesting species. Then we jump into the benthic hydromedusa. This is Tychogastria. There is 3 species of Tychogastria considered valid today. There were a bit more, but they have been synonymized with Tychogastria polaris. The species was originally described from the Arctic, but it's known in the Antarctic waters. And we don't know much about this jellyfish. We know that it can create blooms as well, which is strange for a tiny benthic hydromedusa, but they do. There is a publication in 2017 that they observed huge numbers in the Antarctica, and they were not all sitting on the bottom. They were swimming in the water column. So it seems that it's a species that it's not as it's hyperbenthic still, but it's swimming a lot. And we have already seen that we only in the northern hemisphere we have 2-3 different clades. So we are working on it at the moment, but there is definitely cryptic speciation in there. We were lucky enough to be on board of the Ocean X some months ago. You can see how happy Aino was to go on the submarine. And actually, in one of the dives, that was my dive, it was meant to be, we saw dozens and dozens of Tychogastria CF polaris. We have some evidence that it's probably an undescribed species from the North Sea, and because we've seen that it's not the same as the Arctic one. So we captured a lot of them. We put them in crystals. We took great pictures because, of course, after fixation, they are not going to look that great. And we are working on it, but it looks like a promising finding. Then Dimophyes arctica. The two on the left are from the Arctic. The one on the right is from the Antarctic. Not many morphological difference we can ascribe so far, but it's a tricky species to barcode. There must be some issues with the primers we are currently using. So we aim to sequence the whole mitogenome supported by the Artsdatabanken project NOAA, Norwegian Arctic Hydrozoa. We recently got funding. And yeah, it's our example of holoplanktonic species. So we are crossing our fingers that they will not diverge so much. So it could be potentially the first example of a true bipolar species. We'll see what the molecules say. And last but not least, we have the case of Stegopoma and Tychochina. For those that have been working on taxonomy, this is the perfect nightmare. And I will explain you why briefly. In 2017, Schuchert and collaborators, Aino included, they found out that the polyp of Stegopoma plicatile is actually the polyp stage of the jellyfish Tychochina crocea. What happens? Stegopoma plicatile is known worldwide. It's basically everywhere in the world, from Indonesia, Antarctica, North Sea, Arctic Ocean, basically everywhere. But Tychochina crocea has a very restricted distribution. So we found the paradigm of a widely distributed polyp with a very restricted jellyfish. And because the jellyfish is not found everywhere, then there might be some cryptic speciation there. And it's a nice example of selection acting in only one of the life stages, but not in the polyp, at least with the characters we can recognize so far. Another nightmare, Stegopoma plicatile is the type species of Stegopoma, but Tychochina crocea is not the type species of Tychochina. Another nightmare is they are currently located in two different families. So it's one species located in two different families. It's like having a butterfly in one family and what do you call it? The worm of the butterfly. Yeah, a caterpillar in a complete different family, right? So that's something we have to solve. And then actually the type material of the species it comes from 4 different localities, 2 in the North Sea and 2 in the Arctic Ocean. So that makes the topic even more tricky. What we know so far is that, and as I said, if you check worms nowadays, Stegopoma plicatile is considered an accepted species, and it's synonymized with Tychochina crocea, which is not completely true, because if we keep calling it Tychochina crocea, we will be citing a North Sea species everywhere in the world, and we know that that is far from true. So now we know that other Tychochina species, particularly Tychochina lactea, they have an identical polyp, or nearly identical polyp than Tychochina crocea. And other Tychochina species, whoa, sorry. Hi, oops, can you hear me? Yeah. Other Tychochina species, such as Tychochina hyperborea, it has absolutely nothing to do with Tychochina. It's not a Tychochina. It doesn't even belong to the family, right? So as you can see in these results, this is for Bayesian CO1, but we found the same with maximum likelihood and both for 16S and CO1. You can see that Tychochina lactea has a polyp that is identical to Stegopoma plicatile, but Tychochina hyperborea in the lower part, it's not a Tychochina at all. So we think we found the polyp for Tychochina hyperborea as well, but it's definitely another thing. So take-home messages is that we still have a lot to know about this jellyfish, that we still have to do a better work at studying jellyfish that are not large, they are not conspicuous, because they are also part of the diversity. They also have a key role. They also bloom. Tiny species also bloom, and we really have to do an effort on this. It looks like bipolarity at the species level is probably unlikely, but we are still seeing with Dimophyes what we get in there. And there's definitely a lot of cryptic diversity, but this is our challenge. We care about the identity of the species. I don't need to justify why, because Alan Collins and Mike Dawson already explained why do we really need to make an effort into putting a name or at least a code to the evolutionary lineages that we have, because that have consequences at many levels. And life cycle wise, we have a mess there. We really have to make an effort into try to get this. And if I get 30 more seconds, just a final consideration. On the left-hand side, you have the diversity of gelatinous zooplankton. So most of the gelatinous zooplankton are basically hydrozoa, non-siphonal hydrozoa. And on the right side, we have the tox that we have in the jellyfish bloom symposium, or better called the skyphozoan bloom symposium, right? So I think there is a lot of species we know nearly nothing about, just the fact that they exist, or for many species we still don't even know if they exist. So I think as a community, we really have to center our efforts in underrepresented groups. And with this, thanks to the funding agencies, my collaborators, and thank you for staying.